Namaskar to everyone. Welcome back to my YouTube channel. Today we will discuss about experimental proof for semi-conservative mode of DNA replication. This topic or experiment is studied in a chapter named Molecular Basis of Inheritance in class 2. In this experiment, we come to know about that how two American scientists named Matthew Michelson and Franklin Stoll in 1958 experimentally proved that DNA replicates by semi-conservative mode. So, how this process begins and how this process ends, we will talk about in this video. Okay, so let's get started. First of all, I will talk about our topic, that is experimental proof for semi-conservative mode of DNA replication. In this, what do you mean by the word semi-conservative? So if I wrote here, write here semi-conservative. Here the semi-conservative word is made up of two words. The first one is semi. It means half. And then the second word it is conservative. Conservative means it is to conserve anything. Like as in museums, we see the antique pieces, antique substances or the equipments which are older thousands or 2000 years, where in the museums we conserve them. Okay, so like the museums, like the things which are conserved in the museums, here the DNA, when it replicates, it conserves itself. Okay, so if we talk about this word, so when DNA replicates, it half conserves itself, not whole but half only. Because when the DNA replicates, half the strand or the one strand of the DNA will be new and the half strand will be older. Okay. So before starting our topic, we will look upon that how DNA replicates and in which cell cycle it replicates. Okay. So let's get started. First of all, if I talk about the cell cycle, then we know that in the cell cycle there are two phases. That is the first one, it is the interphase, and then the second one it is division phase. So the interphase is divided into three subphases. That is G1, second one it is S, and third one it is G2. Okay, so here these three subphases comes into a phase that is known as interphase. And then the second phase started, which is known as M or division phase, where the cell divides. Okay, so DNA replication takes place in the S phase. Okay, and how the DNA replicates, we will come to know. Let's assume that it is a cell, and here it is our DNA. So it's normally a double-stranded DNA. So when the S phase comes, then we know that the DNA will divide or replicate. So what happens here? The two strands which are C will separate, like this is one strand and this is the second one. Okay, and then complementary to this strand a new strand will form this kind. Here also same happens, this kind of strand is formed. So what we can see here, that 50% of the DNA is older and 50% is new. So I will write here 50% DNA is older and 50% of DNA is new. And here it also same happens. So in this we can see that the DNA is dividing or replicating by semi-conservative mode, where DNA conserves the parental part and forms newly daughter part. Okay, so how this happens? Now we will talk about the main topic, that is how Matthew Mason and Franklin Stone experimentally proved this experiment. So let's begin. Two American scientists, I will write here. Two American scientists named Matthew Mason and Franklin Stone in 1958 proved that the DNA replicates by semi-conservative mode of DNA replication. Here in their experiment, they chosen a bacteria named Escherichia coli. Okay. Why they choose this bacteria? Even if we know that there are thousands of bacteria in our world, because Escherichia coli, it has a very high reproduction number. It divides or reproduces in every about 20 minutes. Okay, so that's why they have chosen the Escherichia coli. And what they done, that they grown Escherichia coli in NH4Cl ammonium chloride for many generations. So I will draw here, like it choose E. coli, for example, this is the E. coli, and they grew this E. coli in NH4Cl medium. This is our NH4Cl medium for many generations. But what is the point here? That the NH4Cl medium, here the N, this N is not the normal nitrogen which we see or which we study in our normal lives. That is the N14 type of nitrogen. Okay, this is a simple nitrogen which we see or in the books. Here they take a second isotope of nitrogen that have the atomic mass 50. So here I will write here, this nitrogen have the atomic mass that is 15, not 14, because we know that nitrogen has two isotopes. Okay, so what is isotopes? Isotopes, I will say here that if we normally talk about isotopes, that species of an element same number of protons but different number of neutrons. So here if we talk about nitrogen, it has two isotopes. One which has the atomic mass of 14 and one which is the atomic mass of 15 and atomic number of 7, which was used by Matthew Mason's and Franklin Stone. Like this, if we talk about carbon, it has three isotopes. That is the first one which we study, that is have the atomic mass of 12. Then it is the second one which have atomic mass 13, and then it is the third one which have atomic mass 14. Like this, in our periodic table, many elements have different kind of isotopes. Okay, so now we will move forward to our topic. So here they grew up the E. coli in NS share for many generations. And what happens then? The E. coli DNA, or I can say that the N15 type of nitrogen or the isotope was incorporated into the E. coli's DNA. Okay, why? Because for many generations E. coli get nutrition of N15 type of nitrogen or N15 type of isotope only. So if it gets the nutrition of N15, then it will be obvious that the N15 will incorporate into the E. coli's DNA. Okay. So then what they do? That to differentiate the N14 and N15 kind of isotopes, they have a technique, that is cesium, that is the cesium chloride density gradient technique. What is this? We will study. What they do that? They take a test tube, and in this test tube they fill cesium chloride, CaCl. This cesium chloride, what it will do? It will act as a density gradient. I will write here, it will act as a density gradient. What do you mean by density gradient? It means that if I put CaCl in this test tube and then I centrifuge the test tube, okay, then the CaCl will distribute in this test tube according to its density. What I mean, that if I put CaCl in this test tube and then I have to measure a compound or a protein according to its density, then the compound or protein will set according to its density by help of CaCl. For example, if I centrifuge it by taking CaCl in the test tube, then the heavy particles of CaCl will settle down here. The light particles of CaCl will settle down there, and the medium particles which are not too heavy and not too light will settle down at the middle. Then what I will do, if I put a compound in this test tube, then this compound will settle down according to its density. For example, in this compound there are many molecules which are of light density. Then the light density molecules will settle down here, and likewise heavy density particles will settle down here, and the medium type of particles will settle down there. Okay, so in this they do take up one bacteria from the culture plate and then they centrifuge its DNA in the test tube. Okay. So what they come to know that when they centrifuge it in the test tube, then we know that if we take this DNA of E. coli, which is whose DNA is incorporated by N15 kind of nitrogen, then its DNA will look like this. Here, both the strands will be of N15 isotope of nitrogen. And if I centrifuge this nitrogen in density in CSCI density gradient medium, then somewhat this kind of result I have to come to know, that this test tube will have a density band at the bottom part, like here, of N15, because we know that the N15 kind of isotope is heavier, so it will settle down at the bottom part. Okay, then vacuum agents and Franklin install, then they do that, they take up one bacteria from this culture plate, and instead of growing these bacteria in N15 kind of nitrogen, they grow up this bacteria in N14 normal kind of nitrogen plate. Okay, so what they do that, if I talk about the diagram, they take up a bacteria from here, and in this time they grew the bacteria in N14 kind of in ammonium chloride, which contains nitrogen of N14. Okay, so we know that the E. coli divides after 20 minutes, divides after every 20 minutes. So if I look the result, if I write here after 20 minutes, after 20 minutes, what will happen? The E. coli will divide into two because it reproduces. Then, if I talk about these two E. coli and their DNA, so what I come to know that the two strands of DNA will look like this. Here, if this E. coli divides, then first of all, both the strands will separate first. So I will separate here both the strands like this and like this. And then these are the parental strands. It means 50% will be consumed, and the newly formed strands complementing to these two strands will be of N14 type of isotope. Why? Because this time E. coli is grown in the N14 containing nitrogen isotope. So the nutritive element which it will get from the culture plate will be of N14 kind of nitrogen isotope. So I will write here that this time the complementary part of the N15 type, it will be N14. Here it is. And here it is N15. If I centrifuge these two molecules of DNA in the density gradient medium tube, then the results will be of this kind. This is our tube. Here, this time, we know that one DNA molecule contains both the strands, that is the heavy strand, that is N15, and the light strand, that is N14. So it is not too heavy and not too heavy and not too light. So this time they will arrange somewhat around mid of the tube, mid of the test tube. Okay. So we will write here. I will draw here like this kind. This is the N15 and this is the N14. Okay. Then they continue to grow these bacteria in the same medium which contains N14 kind of nitrogen isotope. Then after again the 40 minutes, why 40 minutes? Because here 20 minutes and 20 minutes, if you plus these, it will become 40 minutes. Okay. So what happens here? These two E. coli will again divide. Okay. And when these divide, so we know that the parental strands will be again here. I will write the parental strands will remain assembled here. So I will write here. I will draw here the first parental strand, that is N14. This is N14. Then here it is N15. Then again the N14. And then at the last one that is N15. Now complementary to these four strands will be the isotope N14, because the nutritive medium which is gained by these two kinds of E. coli will be of NH4Cl, which contains N14 kind of nitrogen isotope. So I will write here the N14 kind of nitrogen here. Okay. And if I took these samples into the density gradient medium tube, then the results will look like this. Here, the two strands are made up of N14 DNA, which is the light isotope. So it will settle down around here at the top of the tube. So I will write here N14. And the two strands which contains both type of nitrogen isotopes, that is N15 and N14, it will settle around the mid of the tube. So I will write here. Here it is the N15 and here it is the N14. So what we have come to know, that the DNA divides by semiconservative mode of DNA replication. Here, when DNA divides or replicates, it consumes half of the parental strand and makes half, that is the daughter strand, which is a new strand. So here you can see, and after 40 minutes, E. coli, you can see that half of the parental strand here it is of N14. Here it is also N14, here it is also N14, and here it is also N14. And the new complementary daughter strands, which is it is of different type, which is the nutritive medium. Okay. So hope that today's class was very encouraging, and I hope that you understand my teaching. Thank you.